Hello, my name is Itai Kazaz, and today I will be presenting my research poster on a stability affecting property of the R9L mutation in human gamma-D-Crys protein. The authors of this are myself, Itai Kazaz, Mr. Steen Reich, and Dr. Melissa Kosinski-Collins. The sponsoring institution for this study is the Biology Department of Brandeis University, located in Waltham, Massachusetts. Before discussing my experimentation, it is necessary to give some background information relevant to the research that had been done. This research was inspired by the phenomenon of cataract development in humans, which we observe to be a leading cause of blindness within our species. Cataract formation is caused by the aggregation of a certain protein found in the lens as a result of oxidative stress that accumulates over an individual's lifetime for a variety of reasons. The lens of the eye plays a critical role in the process of sight as it focuses light and images onto the retina in the back of the eye. The protein is called the human gamma-D crystalline, and its aggregation leads to this hindered vision as a result of a pacification, which we call cataract formation. It has been previously studied that this point mutation in the crystalline gene can negatively affect the crystalline protein stability, and so we experimented with the R9L point mutation in this gene to observe its effects on the protein with the hope of better understanding cataract formation. This point mutation induces a switch from the amino acid arginine to the amino acid leucine on an exterior position of the protein. Here you will see an image of the protein itself with the 9-arginine enlarged to show its location on the protein. Now I will spend some time discussing how this study was performed, as detailed through a flowchart. A desired stability-disrupting mutation in the HgammaD-Crys protein was established as R9L. Overlapping primers were designed at the gene level with the sequence that reflects this mutation, and the mutated ChGDine R9L sequence is incorporated into the plasmid and amplified using quick-change site-directed mutagenesis as described. Please note that when I say as described in this presentation, I am referring to the Introductory Biology Laboratory Manual as sourced in the reference section. Wild-type plasmid was removed from the mixture of quick-change products using DPN1 restriction enzymes as described. Amplification of the PQE1 plasmid was verified using DNA agarose gel electrophoresis process as previously described as well. Next, XL10 gold E. coli cells were transferred with the mutated PQE1 plasmid DNA and the culture was grown as described. A minute prep process was followed to purify the mutant PQE1 plasmid from the XL10 gold E. coli cells as described. The sequence of the plasmid was assessed using NIH BLAST to confirm the mutation was indeed incorporated into the mutant genome as described. NEB express IQ E. coli cells were transformed with the plasmid of interest, and a culture was grown as described. The protein was purified from the E. coli through column chromatography, and the purity of the protein was analyzed through SDS page as described. The melting temperature of the mutated crystalline protein was analyzed through a protein thermal shift procedure as described. Likewise, a protein thermal shift was done for wild-type protein as well as for R14C crystalline protein as described. Now that we are clear on how the experiment had happened, I will spend some time analyzing the experimental results as indicated in this section. Here you see Figure 2, titled Melting Point Analysis, which displays the results from a thermal shift assay for wild-type R14C and R9L mutant crystalline. The thermal protein shift assay had been done under standard physiological conditions, which are 10 mmol tris, 10 mmol NaCl, and pH 7.5. This includes error bars to express standard deviations and melting temperature Tm to express protein unfolding. To analyze these results, I will first remind the audience that we had done the thermal shift assay as a means by which to analyze the effect which the R9L mutation has on the stability of the HgammaD-Crys protein, which is why we compare it to the wild-type protein as well as to a protein containing a mutation that is known to be a destabilizing one. As indicated by Figure 2, the wild-type protein has a melting temperature of 85.7 with a standard deviation of 0.64 degrees Celsius. The R14 mutant crystalline had a melting temperature of 80.59 with a standard deviation of 1.21 degrees Celsius, and the R9L mutant crystalline had a melting temperature of 82.95 with a standard deviation of 0.72 degrees Celsius. All in all, this experiment had determined that the R9L mutation being investigated in human gamma-D crystalline protein does indeed have a destabilizing effect on the protein stability as indicated by causing the protein to melt under lower temperatures and physiological conditions. There are many directions for further research based off this experimentation, specifically an analysis of the mutated protein structure with the hope to learn more about its parallel to cataract formation. The protein structure could be better deduced through NMR or X-ray crystallography in future experimentation. Another interesting path that future experimentation could take is to analyze the aggregation of the mutated R9L protein molecules to observe how they interact with each other. I predict that because of this hydrophobic amino acid being placed at the exterior of the protein, that multiple molecules of the mutated protein would aggregate together in a way that parallels cataract formation. Overall, we hope that these findings push the scientific and ophthalmic community towards finding better and more cost-efficient treatments and preventions for cataract formation. I'd like to acknowledge support for this research from Brandeis University's Biology Department through an Introductory Biology Laboratory course offering, and to acknowledge that guidelines for this research have been provided by Introductory Biology Laboratory Manual by Kosinski et al. As noted in references. Lastly, I would like to acknowledge the assistance and collaboration of teaching assistants Tatavik Sarkisian and Abby Daniels, as well as laboratory managers Dr. Kenny Piasta and Dr. Lindsay Marmanish. Here, you will see my references for this experiment as noted throughout the poster presentation. Thank you all for your time, and I hope that this presentation had been informative and entertaining.